Hello, and welcome to part 2 of the lymphatic system and your immune system. We are going to start today with adaptive immunity. The adaptive immunity is how well your body can kind of wage a war and fight against whatever has invaded our system. It could be bacteria, toxins, viruses, even foreign tissues. This is where organ transplants may come into play. But our adaptive immunity is the immune system that comes in, and it's usually pretty specific. We have two different types of specificity when it comes to our adaptive immunity. We have cell-mediated and antibody-mediated. So these lymphocytes are the most abundant of our white blood cells. They are usually the more specialized of the two. We looked at the natural killer cells, which are also lymphocytes. Now we're going to look in more detail at these B cells and these T cells. They are going to develop from the multipotent stem cells and that red bone marrow. T cells, those first types of lymphocytes, they start as pre-T cells. They're going to travel through the blood to get to that thymus. That T is for, again, thymus. That is where they're going to mature and get ready to travel out to do whatever job they need. We gain most of our T cells before puberty because that thymus, as we get older, just tends to atrophy and deteriorate down. Those B and T cells, as I mentioned, are named for exactly where they're going to mature. B for bone marrow, T for thymus. They develop immunocompetence in this bone marrow or this thymus, and it allows them to be able to carry out different types of immune responses. These different immune responses actually create and insert into the plasma membrane these antigen receptors. We talked about antigens a little bit when we talked about red blood cells and blood typing. Now we're going to look at these antigen receptors that are on our on our B cells and T cells, and they're made to recognize specific antigens. Our blood is, again, probably one of the better examples, because some of these B cells and T cells may have antigen receptors that identify our red blood cells as our own. They're the ones who will come in, say if we have type A blood, somebody just gave us type B blood, that type B blood is not recognized, it's going to get destroyed. Our two types of T cells, we have helper T cells, and we have cytotoxic T cells. Helper T cells are also known as CD4 T cells because they have a CD4 protein on them. Cytotoxic T cells are known as CD8 cells because they have a CD8 protein. So our cell-mediated immunity. We have those cytotoxic T cells, and they're going to attack any invading antigens. So anything that comes in that doesn't fit on that antigen receptor, they're going to attack. This allows us to attack against viruses, bacteria, and fungi, even some cancers, foreign tissue transplants. This is how important it is to match up specific tissues when they do organ transplants, because our immune system can come in and attack. So cell-mediated immunity with cytotoxic T cells, it's always cells attacking another cell. Antibody-mediated immunity, these B cells, they came from that bone marrow. They transform into these plasmocytes. They are going to make antibodies or immunoglobulins. These antibodies can actually inactivate antigens. So viruses, bacteria, fungi that are found outside of the cell, we can do antibody-mediated immunity. These antibodies or immunoglobulins that are being secreted by these plasmacytes can bind to these antigens and inactivate those cells. And if they're inactivated, they're essentially useless. They can't come in and invade the body. Helper T cells, they will help with cell-mediated and antibody-mediated immunity. So when specific antigens can enter, or when they do enter the body, originally there's usually only a very small amount of lymphocytes that have that antigen receptor that can respond. Those are helper T cells, cytotoxic T cells, and B cells. Those are the ones that have usually a very small amount of these antigens available and ready to attack. The problem is there's usually more antigens invading the system than there are cells with those correct receptors to inactivate those cells. So what happens when a specific pathogen gets into our body? Once these lymphocytes come across the specific antigen, they find it, they come across it in the interstitial fluid, it cues that lymphocyte to go through what is known as clonal selection. That's where these lymphocytes are going to rapidly regenerate. They're rapidly going to separate out and make more and more of these cells, and they're going to differentiate so they have that specific antigen receptor on their membrane. They can form thousands of identical cells from that one original cell. And this is where we get effector cells, and this is where we get memory cells from. Those effector cells are going to go out and fight. They're going to get rid of that specific pathogen that may have come into our body. These would be those helper T cells, cytotoxic T cells, and plasmocytes that were those B cells. Memory cells, they're made so that they can remember this specific antigen for the next time it comes in, invades our system. So with a rhinovirus, that's one of the types of common colds, and there's hundreds and thousands of different types of rhinoviruses that fit into that category. We come across a specific rhinovirus. We make cells to remember, those memory cells to remember that specific antigen, so the next time we come across it, we can fight it off faster. That's what these memory cells are for. So we'll have memory helper T cells, memory cytotoxic T cells, and memory B cells. These are going to remember that specific antigen for the next time this same antigen shows up in our system again. So here is this entire process. That bone marrow and that thymus are where these different cells come from. We have our cytotoxic T cells, our helper T cells that are going to come out. They come across a specific antigen, these B cells as well. By coming across a specific antigen, they're going to replicate very quickly. Make more and more of these cells so they can go out and wage a war, attack against this specific antigen. But they're also going to make those memory cells so we can remember what happened before, so we don't get sick the second time we come across the same antigen again. This video, you can log into Wiley and click through this link, and you can watch this video on the adaptive immunity. This goes through the lymphocytes and how lymphocytes work. So antigens and antigen receptors. Immunogenicity, that is the ability to actually start an immune response by stimulating specific antibodies. So that proliferation of T cells, B cells, or both. That's where that immunogenicity is. We have reactivity. So the ability of the antigen to react specifically with the antibodies. So that entire microbe or just part of that microbe may act as antigens. These epitopes is just a small part of a large antigen molecule, and it kind of triggers immune responses. So it's just parts of that antigen. So this is an antigen. These different markers on top, or all around this antigen really, are what are considered those epitopes. These epitopes all work to allow specific responses. So most antigens are going to be proteins. They're specific protein markers that are put on cells. Hapten is a substance that it has reactivity, but it lacks immunogenicity. So it can stimulate an immune response, but only when it is attached to a larger molecule. Like the lipid toxin in poison ivy is a good example of a hapten. It can trigger an immune response only after coming in contact and combining with proteins from our body. If you don't have those proteins in your body, these are people who don't react to poison ivy. There are people out there who don't get an immune reaction to it. I've seen a couple people that way, but most people have these proteins that this lipid toxin can come in contact with, and it causes this immune response. Penicillin. This is a medication. It's going to combine with proteins in the body as well, but it's going to form these immunogenic complexes. So this is one way that allergic reactions happen. Somebody who has an allergic reaction to drugs, like different medications, anything in the environment, it attaches to a protein in the body, and then that's what causes the reaction to happen. Antigens are technically foreign substances that are not part of body tissues. So it does happen where the immune system cannot distinguish between self and non-self. We have receptors on our cells that identify our cells as self, but our immune system is not always 100%. And this is where autoimmune diseases actually come in. Your own immune system is attacking cells that your immune system think are foreign. So we've talked about it. It was mentioned in Anatomy 1. We mentioned some of these in Anatomy 2. In type 1 diabetes, the immune system cannot identify those pancreatic isolates that makes insulin. So the immune system comes in and starts attacking these pancreatic isolates that make that insulin. So then those cells can no longer make that insulin. Same thing happens with the thyroid gland. For what's Hashimoto's is one of the types of autoimmune conditions that deals with the thyroid. Your immune system identifies that thyroid gland as non-self, so it starts to attack it. And it starts to deteriorate and break down. So our immune system can actually identify and bind to at least 1 billion different epitopes, these different receptors on those antigens. They can even identify artificially made ones that are made in a lab. So there's a lot of different diversity in these receptors for both B cells and T cells. And it's just by rearranging those epitopes, moving things around just slightly, and that is kind of known as genetic recombination. So the genetic segments are put together in different combinations as these lymphocytes are developing from these stem cells in the bone marrow and maturing in the thymus. So these different genetic recombinations can code for each unique antigen receptor. This is what allows people who comes across specific microbes, they may never react to it because their system, because of this genetic recombination, their system fights it off right away instead of reacting to it. Major histocompatibility complex antigens, MHC, these are considered those self antigens. These are what are used for our immune system to help identify self. These are our cells. We have those transmembrane proteins that go through the entire membrane. These are human leukocyte antigens. So unless you're an identical twin, your major histocompatibility complex antigens are unique to just you. Twins are the only ones that'll have identical ones, as long as you're an identical twin. Every cell in our body is going to have MHC antigens except red blood cells. Helper T cells recognize that an antigen is foreign. So we have two different types of histocompatibility complex antigens. We have class 1 or MHC1. These are right on that built into the plasma membrane of everything except for red blood cells. And then we have class 2, MHC2, and they're on the surface of those antigen presenting cells. Those cells that we mentioned in part 1, where those cells kind of hold up those different antigens and kind of says, hey, who has something that'll fit this? So for our immune system to respond. B cells and T cells have to recognize that this foreign antigen is actually present in the body. So B cells can recognize antigens that are in the lymph, interstitial fluid, or the blood plasma. T cells can only recognize fragments of antigenic proteins. And they need to be presented in a certain way. So this antigen processing is where we take these proteins that are broken down into peptides that associate with these MHC molecules. The antigen MHC complex gets inserted into the plasma membrane of our cells. This is antigen presenting. So when this peptide fragment comes from a self, our own protein, T cells ignore it. They're like, okay, that's ours. We're fine. But if a peptide fragment comes from a foreign protein, something we didn't make, our T cells can recognize it. And the antigen MHC complex is then an intruder. That's when our immune system can come in and fight these cells. Exogenous antigens, they are those foreign antigens that can be found in the fluids, like those extracellular fluids. Some of these are bacterial toxins. These are things that different bacteria release into our system that causes different immune responses. Parasitic worms, people who have a pollen or dust allergy, even viruses. If these viruses have not infected the body cells yet, because viruses will take their genetic material, shoot it inside of our cells, use our cells' components to make or to replicate themselves, and then that cell kind of explodes. But before that virus gets into a cell and starts replicating out, these foreign antigens can be recognized as long as they're in these body fluids. Antigen presenting cells are going to process these different antigens. They're going to present these antigens. So those are those dendritic cells, those macrophages, and those B cells. So these cells tend to sit where these antigens are likely to come out and penetrate these different innate defenses by entering the body. So what happens through this entire process? It's a 7-step process. So there's an ingestion of the antigen. So these antigen presenting cells are going to ingest it. They're going to take it inside of them. They're going to digest this antigen. They're going to break it down into peptide fragments. Because again, antigens are usually proteins, and proteins are chains of peptide amino acids. They're going to synthesize that MHC2 molecule. So that antigen presenting cell that synthesizes that MHC2 molecule, it's going to happen at that endoplasmic reticulum. They're packaged up that MHC2 molecule as well. They're put into vesicles. These vesicles are going to fuse with that cell membrane to present that antigen. There's going to be a peptide fragment that's going to bind to those, or the peptide that's going to bind to those fragments of that MHC2. And then that insertion of that MHC2 complex is going to go into that plasma membrane. So that vesicle now has that MHC2 complex. It's going to go through that exocytosis, and it's going to get completely inserted into that plasma membrane. So those antigen presenting cells, they go straight to the lymphatic tissues because they want to present this antigen that they took in. They ingested, they kind of took it apart into peptides, then they stuck it back out onto that cell membrane. They're presenting it to anything around them when it's attached to the outside of that cell. So they want to present it to T cells. So they have to get into that lymphatic tissue where they can find T cells that may have a receptor that fits that specific antigen. They're going to present that antigen till they find that T cell that fits it. And that T cell, these antigen presenting cells form T cells that intruders, when they're present in the body, they're going to create this action to kill off this specific type of antigen. So this whole process, we get this antigen, we take it in through a vesicle, we break it apart, we get this major histocompatibility complex 2 molecule that comes into this membrane. It's going to pick up these antigens and present it. It's going to come and bind back out to that cell membrane. We're going to present it out and say, hey, who has this? Who works with this? An endogenous antigen. These are different antigens that we can find inside of the cell. Viral proteins. Viruses, they infect cells, they take over that cell's machinery, uses everything up. Toxins from bacteria. Even abnormally synthesized cancer cells. Our system can still find these that are inside of the cell. So endogenous antigens, I like pictures. This antigen that is inside of the cell, it's going to get broken apart. It's going to be taken up by that major histocompatibility complex 1 molecule. That's going to come in and take this antigen, and we're going to attach it right out to the outside of that cell membrane here to say who fits this, who can work with this. We need to kill this off. This is inside of my cell. Help. Cytokines. These are protein hormones. They can stimulate or inhibit cell functions. So cells growth, they can inhibit that cell from growing. They can inhibit that cell from differentiating. They can also stimulate it to happen sooner than it should. Lymphocytes are going to secrete cytokines, so do antigen presenting cells. These cytokines can cause the proliferation of those progenitor blood cells. So we can start making more red blood cells. They can be involved in those innate defenses or those adaptive immune responses. So these are all the different types of interleukins. Most of them we don't really talk about, or the different types of cytokines. A lot of them are interleukins, I should say. Switch that around. These interleukins can cause different things to happen. Like interleukin 1 can cause a fever. Interleukin 2 can cause different cells to proliferate, immune cells. So cell-mediated immunity, we need cells to become activated first. So we need the activation of a small number of cells before we can actually go through with an immune response. So we need a small number of T cells with this specific antigen. So once these T cells have been activated, because they usually sit inactivated until something comes along that they can work with. They come through and become activated. They're going to go through that clonal selection where they go through and replicate themselves very quickly to make identical clones of themselves. Some of those T cells become those effector cells that go out and fight. Other cells become memory cells that are going to remember. The effector cells of those T cells eliminate the intruder. Whatever microbe, bacteria, whatever has invaded that cell comes right back and says adios. So T cell receptors. So antigen receptors are usually on the surface of these different T cells. So they're going to recognize this specific foreign antigen that these major histocompatibility complexes are presenting. When that antigen enters the body, again, we usually have a few T cells that are going to identify it. These antigens, antigen recognition is going to involve either the CD4 or the CD8 cells, those different T cells. The recognition is going to cause the activation first of that T cell. We need to activate that T cell before we can start to replicate it. So when it becomes activated, it's only going to get activated if it can bind to that antigen in the right way. That causes co-stimulation. So those cytokines, interleukin 2, can get activated. So there's technically two different signals that are needed to activate this T cell. So it's like inserting a key into a lock. A certain key will only open a certain lock. That is how these T cell receptors work. If it's not an exact fit, it's not going to activate that T cell. So these different co-stimulators affect these different T cells. So recognition without co-stimulation leads to what is known as a state of, or a prolonged state of inactivity or anergy of both T cells and B cells. So once a T cell receives these different signals, it is activated. So it needs to fit in and it needs to be turned, like a key in a lock. So most T cells have this CD4, that displays CD4 are helper T cells or CD4 T cells. So we have these inactive helper T cells. When they recognize these different fragments from the major histocompatibility complex 2 or from antigens that are found outside of the body, these CD4 proteins, the helper T cell and the antigen presenting cell, they're going to all interact with each other. This co-stimulation occurs, and then the helper T cells become activated. It's kind of another one of those chain reactions that happens. There's a lot of chain reactions in your body. So after that T cell becomes activated, it goes through that clonal selection where it's going to make clones of itself. So within hours of that cell being stimulated, we have active T cells that are going to secrete these cytokines, mostly interleukin 2, that are going to cause an immune response. We get those memory T cells that come in. So memory T cells of the helper T cells, they're not active. But if that same antigen comes back in the future, comes back again, these memory T cells can proliferate. They can differentiate, and they can turn into active T cells to fight this same antigen again. So through this process, we have this antigen presenting cell presenting out this antigen saying, hey, who fits into this? In this, putting that lock in that key, turning it activates that cell. That cell is going to make many, many clones of itself to go out and fight it. Some will fight, some will be our memory cells. We have these T cells that display that CD8 protein. Those are those cytotoxic T cells, or those CD8 T cells. These are the ones that bind with the major histocompatibility complex 1. So they have some sort of foreign antigen inside of their cell. The CD or the MHC, major histocompatibility complex 1, shows these antigens from what they found inside themselves. These can be body cells, tumor cells, even transplant cells. So for recognition, these TCR cells, those cells that are presenting, and these CD8 proteins come together and it'll activate. So after this recognition, these cytotoxic T cells become activated, and they actually require stimulation by interleukin 2 or other cytokines by these helper T cells. They also bind to these same antigens for these cytotoxic T cells to become activated. It takes one more step to cause these cells to become activated. So once this happens, once they become activated, we're going to become bound to different copies of these antigens, which activates these cytotoxic T cells. They go through those clonal selection, just like our other T cells do. We're going to make active cytotoxic T cells. We're going to make memory T cells. Active T cells attack. Memory T cells, they're our memory of what we came across the first time. So for these cells to be activated, they have to fit into the receptor, turn that lock and key. We need that interleukin 2 from that helper T cell to come in and also stimulate, to activate these cytotoxic T cells into proliferating, to making several clones or many, many clones of themselves. So once we've activated these cells, they've gone through that clonal selection where they multiply and make many, many more of them. These cytotoxic T cells, they're the soldiers. They're at the front. They fight these invaders however they can. They're going to leave those lymphatic organs. They're going to go through that lymphatic tissue. They're going to go through that lymphatic system and find the cells with these antigens. They could, these different target cells, cancer cells, transplanted cells. They're going to find and attach to these target cells. They deliver what is known as a lethal hit that is going to kill that target cell. Cytotoxic T cells, they kill infected cells, kind of like natural killer cells do. But these cytotoxic T cells have receptors that go for a specific microbe versus natural killer cells just attack. Doesn't matter what antigen is there. These cytotoxic T cells have to find this specific receptor to be able to attach and kill. That's what this does. So these cytotoxic T cells have two ways. They can use receptors on the surface where they can bind to this infected cell, and then they can release granzymes, which are proteins that they're protein digesting enzymes that are going to trigger this kind of apoptosis, cause that cell to go through that programmed cell death. They can also bind to these cells and they release proteins, perforin and granulosin. Perforin creates all these channels that causes that fluid to come into that cell to burst. Granulosin is going to go through these channels. They're going to destroy the microbe by making holes in that microbes in their plasma membrane. They can also release lymphotoxin, which causes enzymes in that target cell to get activated, which causes that target cell's DNA to actually fragment. As soon as that DNA breaks apart, that cell dies. They can release gamma interferon, which causes and it activates phagocytic cells, and it kind of prevents those phagocytes from leaving that area. So once these cytotoxic T cells get attached to their target cell, or they're going to secrete one of these substances in, and then they're going to detach. They're going to go to another cell, attach to another cell, and destroy that next cell. Because usually when these microbes get in, they come in large numbers. They're going to invade several cells. So our immune system, these cells don't die with the cell that they are attacking. They keep moving from cell to cell, killing off multiple cells at the same time. So they go from one cell to the next cell, sending in would be granzymes, granulosin, perforin, causing these cells to die in one of many ways. And then these phagocytes are at the other end. As these microbes are leaving the cell because that cell died, those phagocytes are taking them in and killing them. So our immune system can kind of survey what is happening around the body. Normal cells can sometimes transform into cancerous cells. And when that happens, it actually displays what is known as a tumor antigen. The problem, they very rarely display on a healthy cell. We really only see them on these different cancerous cells. So if our immune system is kind of surveying, going around, checking what is happening, they may recognize this tumor antigen as its non-self. And it can destroy any cancer cell that has this antigen on it. These are carried out by those cytotoxic T cells, those macrophages, and those natural killer cells. They are actually the most effective at actually getting rid of cells, tumor cells, because of viruses that cause cancer. We have plenty of viruses out there that cause cancer. These cells can recognize and get rid of these cells that are invaded by these different viruses. It may happen that somebody's had an organ transplant. What happens when somebody has an organ transplant is they have to take immunosuppressive drugs. These immunosuppressive drugs prevents their immune system from coming in and attacking this new organ that was just put into their system. That is what's called as transplant rejection. They're trying to prevent that by giving them these immunosuppressive drugs. These people, unfortunately, have an increased risk of virus-associated cancers because they're suppressing their immune system so it doesn't find this new organ as foreign, but it also prevents that immune system from going out and finding possible cells that are invaded by viruses that could eventually cause cancer. So this antibody-mediated immunity, an antigen gets recognized and bound. These helper T cells co-stimulate those B cells. Those B cells and T cells proliferate and differentiate. They clone themselves, and they're able to eliminate the antigen, and essentially that what is causing that antibody-mediated immunity to come in and work. So during the activation of B cells, an antigen is going to bind to B cell receptors. They're similar to antibodies, and they get secreted out by those plasmacytes. B cells respond to unprocessed antigens presented in lymph plasma or that fluid. And their response is a lot more intense. So these antigen processing B cells, what they do, this antigen is taken inside of that B cell. It is broken down into those fragments, and it's combined with that MHC2, and it is moved to that plasma membrane. Helper T cells are going to recognize that antigen. They're going to co-stimulate more B cells to divide, proliferate, differentiate. These helper T cells produce that interleukin 2 that co-stimulates and activates those B cells. So those B cells can undergo that clonal selection, and that results in the formation of B cells that become plasmacytes and those memory B cells. Those plasma cells secrete out those antibodies. After a few days, those plasmacytes secrete out hundreds of millions of antibodies each day for about 4 to 5 days until that plasmacyte dies. So those antibodies are going to travel throughout the body, that lymph plasma, that blood plasma, to wherever this antigen has come into the system. There's interleukin 4 and interleukin 6 that are produced that help by helper T cells. They help to enhance that B cell proliferation and B cell differentiation. That B cell differentiates into plasmacytes, which will then secrete out antibodies by those plasmacytes. We keep those memory B cells. They don't secrete out antibodies, but they can proliferate and differentiate into those plasma cells when that same antigen comes back. So these different antigens, they can stimulate different B cells. These B cells have to differentiate into plasmacytes before they can secrete out these antibodies. So all B cells have a certain clone, or they can secrete out only one type of antibody. So each specific antigen can only activate the B cells that can secrete out that specific antibody for that specific antigen. So these antibodies that are produced by a clone of plasmacytes, they're going to travel throughout the body into the circulation. They're going to bind with these antigens, and they're going to work to kill things off. So we have this inactive B cell. We find this antigen that attaches to this major histocompatibility complex. These antigens fit in, which causes helper T cells to help co-stimulate so these B cells can replicate. These B cells are going to secrete out millions of these antibodies, but we create those memory cells that are going to remember those. So an antibody specifically, antibodies can, again, they combine with these epitopes that are on these antigens. And it kind of triggers production. It's another one of those lock and key. They only fit specific things. Antibodies belong to this group that are known as immunoglobulins. They're glycoproteins plus globulins. They usually have 4 polypeptide chains to them. They can be light, they can be heavy. We have variable types, but the main important thing is we have the different types of antibodies. We have 5 different classes: IgG, IgA, IgM, IgD, IgE. Those are immunoglobulin G, immunoglobulin A, immunoglobulin M, immunoglobulin D, immunoglobulin E. If you have a high number of IgMs in the body, that's because of a recent exposure to something. So these different immunoglobulins have different binding sites. Again, each binding site is specific to its specific epitope. Everything is different. We have neutralizing antigens. And neutralizing antigen, so it's the reaction where that antibody, an antigen, with that antigen, they block or it neutralizes those bacterial toxins. It can also prevent viruses from attaching to our specific cells and invading our cells. We have a mobilizing bacteria, so if these antibodies form against those specific antigens that are found on the cilia or the flagella of bacteria, because bacteria usually have one of those 2 things that help move them. It can cause that bacteria to lose their motility. And when they lose their motility, they can't spread from cell to cell. We have agglutinating and precipitating antigens. When that antibody and antigen, that reaction, can cross-link with pathogens, and it can cause agglutination. So these phagocytic cells, they ingest the agglutinated microbes and break them down. Activating complement. So they initiate this classical pathway of the complement system. And then we have enhancing phagocytosis. And this is where this that region on that antibody, it's going to act as a flag that causes these phagocytes to come in. So once these antigens bound to this antibody, these antibodies enhance that activity of the phagocytes. So ingesting them, coating them, whatever it takes, so these phagocytes can come in and completely ingest and break them down. There's a lot of different immunoglobulins. They cause different kinds of reactions. So this complement system is another defense system that has over 30 proteins in it that are made in the liver, and then they're found circulating throughout the blood. These different proteins destroy microbes by causing this phagocytosis. They cause inflammation in that area, and they're also going to prevent too much damage to the body tissues. We tend to use uppercase letters when we're talking about complement. We have complement 1, complement 2, all the way through 9. These are different proteins that are technically they're inactive. They only become active when split by enzymes. So enzymes come in and break them apart. Again, they're going to act as in a kind of a cascade. So one reaction triggers another reaction, which triggers another reaction, which triggers another reaction, and so on. So each time we have a reaction, more and more product is formed. So it helps to amplify these effects. So this complement activation, it's got 3 different pathways. They all activate C3 protein, and that begins this different cascade reaction. And that brings phagocytosis, cytolysis, and inflammation. So cells ingesting, cyto is cell, lysis is breaking it apart, and then inflammation. So these C3 is going to split the CA or C3A, C3B. Here's a better view. So this complement protein splits off. It can go over and activate phagocytosis. It can also activate those mast cells because those mast cells are going to secrete out that histamine that causes that inflammation to come into the area. C5, that complement 5 protein can split off and activate those mast cells. C6 causes C7, C8 to activate, which causes C9 to activate. They all attach into that cell membrane, which causes that cell to lyse. So we have different pathways that can activate these. We have the classical pathway, when an antibody binds to those antigens. We have an alternative pathway. We don't need antibodies to cause this one to activate. And we have the lectin pathway, where macrophages digest microbes. They release out chemicals that cause the liver to produce lectins. Lectins can bind to different carbohydrates on these microbes and cause the activation of C3. Immunological memory. Our immune system remembering everything it has come across. It is because of those antibodies and those lymphocytes that are made during that clonal selection. Cell-mediated and antibody-mediated responses are very quick. And after the second or subsequent, so 3rd, 4th, 5th exposure to an antigen, it's even faster. So usually we only have a few cells with that specific binding site. So it takes several days to build up that intensity to kill off those microbes, those pathogens that have gotten into our system. That's why the first time you come across some sort of pathogen, microbe, you usually feel sick for some time before you start to feel better. So what is happening during this time is we're finding the correct cells to activate that match these specific binding sites. We're activating those cells. We're going through the entire clonal process where these B cells and T cells are replicating rapidly, but it takes time. So eventually we have thousands of memory cells. So after this initial encounter, the next time these cells can proliferate very fast. It can happen within hours. So if it's happening within hours, you may not even know that this virus has invaded your system because your immune system has already gotten rid of it before it's gotten a chance to make you sick. We use what are known as antibody titers. If we want to know what memory cells you have of what you've come across, we can run titer tests to see if you have these specific antibodies that are found in the serum of your blood. So after that initial contact, no antibodies are usually present it takes time for those antibodies to show up. It takes days. We will see a rise in IgM, immunoglobulin N, and then we start to see that rise in immunoglobulin G. And then there's usually a decline. That's in our primary response. What I wanted. The second exposure, the same pathogen gets into your system. Well, within a few hours, we see a rise in IgG, we see a rise in IgM. After that second response, the amount of antibodies remains a lot higher. That is that secondary response. These primary and secondary responses tend to occur in those microbial infections. So whenever you recover from some sort of infection without using medication, it's because of that primary response. If that same microbe comes in, that secondary response could be really quick to prevent you from getting sick again. So these are the different types of immunity that we have: the naturally acquired, active, naturally required, passive, artificially acquired, active, artificially acquired, passive types of immunity. T cell recognition and self-tolerance. So to function properly, your T cells have to have two different traits. They need to be able to recognize your own major histocompatibility complexes, MHCs, those proteins, so that's self-recognition. And they have to lack reactivity. So they must not react to your own cells. That's that self-tolerance. These T cells can identify your own cells, so they're not going to activate and react to your own cells. B cells also have self-tolerance, but if they lose that self-tolerance, that's what causes those autoimmune diseases that we mentioned earlier. T cells in the thymus will develop this capacity for self-recognition. It's known as positive selection. So some of these pre-T cells express T cell receptors, and they're going to interact with these self-major histocompatibility complex proteins that are found on these cells in the thymus. It allows these T cells to recognize that major histocompatibility part of this antigen, MHC antigen complex. Those are the T cells that will survive. Immature T cells that cannot react or that not interact with those epithelial cells, those thymic cells, they're not able to recognize self. They're the ones that undergo apoptosis. So if they cannot recognize self cells, these different T cells, they're going to die. If they can recognize your own self cells in the thymus, that's what allows these cells to live. This self-tolerance occurs through weeding out, or that negative selection. So T cells are going to interact with nodular dendritic cells. And these T cells with receptors that recognize these self-peptide fragments of other self-antigens are eliminated. T cells selected to survive, they're not going to respond to these self-antigens. So we use this negative selection. So we use deletion, where those T cells go through that apoptosis, or we use anergy. They remain alive, they're just unresponsive. They're not going to work. So once these cells come out of the thymus, they can still encounter things that they don't know. And they can still become anergic. They can also undergo deletion after leaving the thymus because they can still come across things that they're killing off things that they really shouldn't. B cells are going to develop that tolerance also through deletion and anergy. Those B cells, again, come from in that B for bone marrow. There's cells that are going to exhibit antigen receptors. If they recognize common self-antigens, they are deleted because we don't want them to identify those common self-antigens. Once they get released into the blood, anergy appears. That's how they seem to die. They just get inactivated. So when these B cells come across these antigens that are not associated with antigen presenting cells, that co-stimulation that they need to replicate through those T cells doesn't happen. It can be missing. And if that happens, those B cells can become anergic. They get inactivated completely, so they can't ever work, instead of becoming activated, which can create a problem. So these B cells in that bone marrow, if they start to recognize self cells, they get deleted. Once mature, they go out and through to circulation. Through co-stimulation, they will make plasmacytes. If there's no co-stimulation, they become inactivated for life. So here's all the different types of antigen presenting cells, all the different types of lymphocytes and what they do. Stress. Stress and your immune system. They tend to work together. Psychoneuroimmunology is a field of study that looks at that link between the nervous system, the endocrine system, and your immune system. So they're going to look at your thoughts, your feelings, your mood, how you feel, like happy, sad, even your beliefs. Things that you believe in can influence your health and how your body fights diseases. That hormone cortisol that's secreted in that stress response, cortisol can inhibit your immune system from working properly. There are people that are resistant to the negative health effects of stress. These people, interestingly, experience a sense of control over the future, commitment to their work, and expectations of generally positive outcomes for themselves. And they have this social support system in place. So, according to psychoneuroimmunology, if you want to increase your stress resistance, you need to have an optimistic outlook on your life. Get involved in your work. And then you need to build good relationships with others. You need to get enough sleep and relaxation. You need to take time to just relax, kind of veg out. Skipping sleep maybe can give you more time to do other things, but in the long run, you fall further behind on things you were trying to do, especially if you end up getting sick. It can keep you out of commission for days, and it can back you up big time. Lack of sleep, you lose your concentration. It starts to cloak creativity. You're not as creative in your thoughts. But the problem is sometimes even if you make enough time for sleep, stress can cause insomnia where you have a hard time sleeping in general. If you can't sleep, the number one thing to do is start looking into how do you manage your stress? What kinds of things do you do before you go to bed? These are different ways to try to help your immune system. No matter what you do, aging is still going to play a role. Your immune system is not as efficient because it increases that susceptibility to infections and malignancies, those different tumors. You have a higher chance of producing these autoantibodies. You have that decreased response to vaccines. Your immune system just decreases in general. Doesn't function as well as it did when you were younger. So your lymphatic system, it plays a role in every system in your body, whether it's your skin, like your skin, the lymphatic tissue provides this IgA antibody in sweat. Your skeletal system has lymphatic vessels around it. Your muscular system has lymphatic vessels, and your muscular system works as a pump. That endocrine system, those hormones, those cytokines, respiratory, digestive, urinary, genital systems, all of these different systems are going to deal with your immune system. So let's go through some different diseases here. HIV and AIDS, or acquired immunodeficiency syndrome. That's what AIDS stands for. A person who has acquired immunodeficiency syndrome, these people tend to experience lots of different types of infections because their immune cells are invaded by this HIV virus, this human immunodeficiency virus. AIDS is transmitted, or HIV is transmitted through blood and some bodily fluids. That's why Whenever you are working with any kind of bodily fluids or blood, you always need to wear some kind of protection, some kind of gloves. Because AIDS isn't the only thing that can get spread this way. We'll talk about more throughout the semester. And it is spread the best when people are doing things that are going to exchange bodily fluids, such as unprotected sex, anal, vaginal, or oral sex, all three of those. Direct contact with somebody's blood, from an HIV-infected mother to her baby. It is a very fragile virus, so it doesn't live outside our body. So that is one good thing. So you're not going to get it from touching a table, touching the soap dispenser that somebody just used. You're not going to get it that way. You can only get it through sharing bodily fluids. It is a double-stranded RNA virus. It has different enzymes. It has this capsid. It has a membrane. It is considered a retrovirus because that genetic information is RNA. It replicates by entering our cells using our cells' materials. The problem is the cells that it's going to invade are helper T cells. So through these helper T cells and how they can replicate so easily and fast for our own immune responses, HIV can make up to 1 billion copies of itself every day. That is just crazy how many of these cells can be created so fast. So our body defenses can kill these infected cells, but because they replicate so fast, we just can't keep up with it. That is how HIV starts to spread throughout our body, because we just can't keep up with how fast these cells are replicating. So once people get infected, they tend to get like flu-like symptoms, fever, rash, fatigue, headache, joint pain, sore throat, swollen lymph nodes. And they'll experience that for a little bit, but about 3 to 4 weeks after the infection, these plasma cells start to secreting antibodies against HIV. We can detect them in the blood. So you can actually screen for HIV blood tests for HIV after about it does take 3 to 4 weeks before you can actually detect it. That's the kind of the scary part. It takes time before your body can detect that it's actually there. 2 to 10 years, HIV destroys enough of those helper T cells that people start to experience this immunodeficiency, which means other viruses can come in and attack, and your immune system does not have a way to fight it off. HIV can start to infect the neurons in the brain, which causes memory problems, can cause visual disturbances. These other viruses that come in and invade the system are known as opportunistic viruses. A lot of times people with HIV and AIDS, this is what they die of, is opportunistic infections, because their immune system cannot fight it off. There's different drugs and treatments that help kind of suppress the HIV virus. We have nucleoside reverse transcriptase inhibitors, try to prevent that RNA from replicating itself. We have non-nucleoside reverse transcriptase inhibitors. And these are drugs that they're trying to help prevent this HIV virus from replicating so much and causing problems throughout the body. Another immune problem is allergies. This is when somebody is overly reactive to some sort of substance that most people tolerate very well, tolerate just fine. When an allergic reaction occurs, there is tissue damage that happens every time an allergic reaction occurs. The antigens that cause an allergic reaction are known as allergens. There are certain foods that people are allergic to. Nut allergies have been pretty prevalent. Shellfish, antibiotics. There's certain antibiotics that people are allergic to. There's a lot, I know quite a few people allergic to penicillin. Venoms, cosmetics, poison ivy. As I mentioned before, not everybody responds to poison ivy. Only certain people do. Pollen, iodine-containing dyes. Some people are allergic to iodine. I break out into hives to iodine anytime it touches my skin. Even certain microbes people can actually have an allergic reaction to because they're secreting out like certain toxins. There's 4 different types of allergic reactions or hypersensitivity reactions. Type 1 is that anaphylactic. Anaphylactic reactions are not always what people think they are, where that throat closes up, where they can't breathe. It can include hives, itchiness, flushing, faster or slow heart rate, low blood pressure. It can actually cause lightheadedness, headaches, anxiety, confusion. These are different signs of anaphylaxis. Shortness of breath, wheezing, coughing, pain when swallowing, even abdominal pain, diarrhea, vomiting, and loss of bladder control. These are all different signs and symptoms of anaphylaxis. There's cytotoxic. That's a type 2 hypersensitivity. Type 3 is an immune complex, and type 4 is cell-mediated. This type 1 anaphylactic, it is the most common type. Within a few minutes of exposure, someone can have a reaction. And here's the interesting thing. These allergies can develop at any time in your life. Like somebody who now has a shellfish allergy, myself as an example, I used to be able to eat shellfish just fine. I went about 3 or 4 years without eating shellfish, was exposed to shellfish, and had an anaphylactic reaction to it. So my body just developed it over the years. So people are going to they can produce this IgE antibody that it binds to mast cells, it binds to those basophils, that lets out that histamine, that lets out those granules that causes that inflammation, that redness, sometimes that swelling. So the next time that allergen enters that body, that IgE, we've already recognized it because it entered that body the first time. It entered and attached to those mast cells, those basophils. So the second time it comes in, it causes that allergic reaction. So that histamine is going to release prostaglandins, leukotrienes, kinins. These cause vasodilation of those blood vessels. They could close off. It could increase that blood vessel permeability. Can contract the smooth muscles in the air, in the lungs, which causes decreased ability to breathe. It also can increase mucus secretion. That's where people may have difficulty breathing. It can cause that runny nose. Anaphylactic shock. It's kind of accompanied by this shock where this person has vasodilation. So they're losing a lot of fluid from the blood because their blood vessels have gotten so large. They can start sweating. It can be life-threatening. EpiPens, that is injecting epinephrine. into the system because it vasodilates different areas. It's going to vasoconstrict. It's going to cause these reversal of this anaphylactic type of reaction. The problem is these EpiPens aren't long lasting. They don't last very long. So it gives you time to get somebody to get help. Type 2 reactions are usually caused by IgG or IgM, and it's usually against antigens on a person's blood or tissue cells. Type 3 reactions involve these antigens with antibodies IgA or IgM and that complement system. So somebody who has rheumatoid arthritis, glomerulonephritis, we'll talk about that more in the renal system. Type 4 is a delayed hypersensitivity. So people who experience different reactions, but it takes 12 to 72 hours for that reaction to happen. This is the time when people don't always realize that they're having an allergic reaction to something because it could have been something that happened 3 days before that is causing this reaction to happen. Poison ivy. Poison ivy takes time to show up. Tuberculosis. This is why when you get TB tested, if you do those skin TB tests, those skin TB tests are checking for this delayed reaction. It takes 24, you have to read it within 48 to 72 hours because it's looking for this delayed reaction. Some people can have these delayed reactions to food that they eat. Something topical they put on their skin. It can happen to a lot of different things, and people don't always put 2 and 2 together on these type 4 reactions because it could have been something that happened 3 days before that is actually causing this reaction to happen. Most of my skin reactions take at least 24 to 48 hours to show up. Like the iodine that I mentioned that I was allergic to a couple slides ago, that one's about 18 to 24 hours after exposure. I'm full on in hives wherever they put that iodine. And then there are autoimmune diseases. That's when our immune cells no longer can identify self. They don't have that self-tolerance. They can't identify self versus non-self. So they start attacking their own tissues. It usually starts in early adulthood, and unfortunately, females suffer it more often than males. These cells, that anergy and that deletion that's supposed to happen does not happen. So that's where that type 1 diabetes, multiple sclerosis, where your immune system attacks that insulation on your neurons, whether it's those oligodendrocytes or those Schwann cells. Rheumatoid arthritis, your immune system is attacking your synovial joints. Addison's disease, that's where your immune system is attacking your adrenal glands. Lupus affects a lot of different systems. And Hashimoto's, your immune system is attacking your thyroid gland. All of these are just because your immune system can no longer identify self from non-self, and it causes your immune system to start attacking your own body's cells, which starts breaking down different tissues. Well, this is everything for this chapter, chapter 22. Thank you very much for listening, and I will see you again in the next section. Thank you. Bye.